Hello, welcome to this lecture on biomathematics. We have been discussing statistical thermodynamics of various biological systems. So, today we will continue discussing that. So, the module, the section is statistical thermodynamics of biological systems. We will be, in today's lecture specifically, we will be discussing thermodynamics of protein organization along DNA. So, what do I mean by thermodynamics of protein organization along DNA? You all know that in cells, DNA is not just a simple polymer. There are many proteins that is binding and dissociating and is bound onto the DNA, and many proteins will have to do various functions by binding onto DNA. So, what we want to understand is a simple a system similar to, we would not go into the complexity that we see in biology, but we will take a simple system that we can think of as an in vitro system, and we ask the question, using thermodynamics, can we say something about protein binding and dissociation. So, one aspect of this is again a very wide, there is large amount of, it is like very big topic, that proteins binding to DNA, but we will only ask a very simple question in this, a very limited, our purpose is very limited, we will ask only one simple question. Imagine that you have a DNA of particular length, and there are, let us say, some number of binding sites, let us say n binding sites, and there are some number of proteins available. Using thermodynamics, can we predict how many proteins will be bound onto the DNA? If you have, let us say, 10 binding sites and 7 proteins are available, can we say all the 7 will be bound or only 5 will be bound? Can we say, can we predict something about this? And the answer is yes, we can predict, and thermodynamics will, the statistical ideas that we discussed will tell you that how to predict this. Okay, so let's think about it. So, have a look at here, what we are going to discuss is, so we have, imagine the DNA as a line like this. So, we have a certain DNA of interest, and we are thinking of in vitro situation. Let us say somebody is doing an in vitro experiment, where they take a DNA of particular length L. So, L is length of DNA. So, you take, let us say 1 kb, 2 kb or few MB DNA, and imagine that there are certain binding sites along the DNA. So, let us say there is a binding site here, there is a binding site here, and there is a binding site here. There are 3 binding sites, and there are various proteins in the solution. So, this is the in vitro situation we are thinking about. So, there are, you have a DNA of length L, and you have 1, 2, 3 here, like, so let us say there are n binding sites. So, this proteins bind to some specific sites, and there are n of them, such, so here n is equal to 3, and then you have, let us say, m proteins. So, here I have like 4, these circles I draw are proteins. So, let us say m proteins in the solution. So, this is what somebody doing an experiment, they take a DNA of length L, and there are n binding sites and m proteins in the solution. Now, the question is, we are asking is, how many proteins will be binding to this? Will all these 3 sites will be occupied, or will only one be occupied, or only 2 will be occupied, or what is the probability that each of this, like it's everything is some you can think of as probability, or what is the equilibrium state at in equilibrium, that is what is the most likely state, that is will it be all will be bound on this, or like how many of them will be occupied. So, the question we are asking is essentially what percentage of the sites, what fraction of the sites will be occupied. So, let's say, right, if there are n binding sites and m proteins, in at equilibrium, how many of the proteins will be bound onto the DNA? How many of the proteins will be bound onto the DNA? So, this is the question we are asking, and we can at the end of this lecture there will be an answer. This many, what will be the answer will be how there will be a certain number like 20%, 30%, 50%, 80% of the sites will be occupied by proteins. We can answer this question. So, to look at this, when we think we are discussing statistical thermodynamics, so some ideas from statistical thermodynamics has to be used here. So, we discussed that equilibrium is decided by the minimum of free energy. You have to find the free energy and see, look at where is the minimum of the free energy, and that will be the equilibrium state, that will be the stable equilibrium state. So, we want to calculate free energy of the system, that is the way to go ahead. So, if we can calculate the free energy of a protein DNA system, we will be able to tell something about this, we will be able to answer this question. So, let us think about the free energy of the system. So, before thinking of free energy, what happens when proteins bind to DNA? So, let us look at that a minute. So, typically DNA is something like negatively charged, and protein typically will be like positively charged. In this case, let us say positively charged. So, when they bind, there will be an attraction between this. So, if you think about this energy, this let us say this interaction energy, the proteins will be, so let us say there is an electrostatic attraction and the energy is q square by 4 pi some all the constant, let me write k into r. So, our r is the distance between these two charges. So, this is the Coulomb energy. So, if we have the Coulomb energy, this will tell you that when the r is, and there is a minus sign, there is a plus charge and minus charge. So, there will be a minus sign there. So, what does this tell? This tell that if you plot this E c versus r, Coulomb energy versus r, what you would get is that some curve like this. As r goes to very small, as r goes to 0, so this is 0, the energy will decrease, because as r goes to 0, this will go to minus infinity. So, the energy will be minimum, the Coulomb energy will be minimum when they bind. Binding means they come together. So, when this protein binds to the DNA, the r will decrease and the Coulomb energy will be minimum. So, according to the Coulomb energy, they would want the proteins to bind, so that the energy is minimum. In that sense, if you think of that, all the proteins should bind. Okay, so, let us think about, so there is some energy associated with binding, and it involves Coulomb energy plus some other interactions like self avoidance interactions and so on and so forth. So, this will be a little more complicated energy, but let us say when one protein binds to a DNA, the system gains an energy, let us say minus epsilon kBt. So, let us say this is minus epsilon kBt. So, what is this? So, this is energy gained when one protein binds to DNA. So, this is certain epsilon can be some number 1, 2, 3, 4 or 1.5, 3.5. So, kBt is a unit of energy. So, the kBt is 10 power minus 21 joule. So, certain amount of joule of energy will be gained. So, when a protein binds to DNA. So, when a protein goes and binds, certain amount of energy will be gained. So, this is the energy gain. So, if there are two proteins bind, the energy gain will be minus 2 epsilon kBt. So, let us think about it. So, when there are two proteins bind, the one protein minus epsilon kBt is the energy gain, two proteins minus 2 epsilon kBt. So, similarly 3 protein minus 3 epsilon kBt. There are m proteins. The energy gained is minus m epsilon kBt. Okay. So, this is the energy gain. So, what are we writing here? So, let us say write. So, if we plot, so let us plot for a minute the energy gain. So, the energy gain by protein binding versus number of proteins m. So, the more if there is just one protein is just, so this is 0. So, let us say there is some certain energy gain and the curve will be, we said that the curve will look like minus energy is minus m kBt, minus m epsilon kBt. So, epsilon is a number, kBt is a number. So, epsilon is a positive number, kBt is also a positive number. So, this is like y is equal to minus some constant m times x, x is m here. So, this graph will look like this. So, something like this, the graph will look something like this. So, what does that mean? The more the number of the proteins bind, the energy is less and less. So, if there are m, so this is m is equal to 1, m is equal to 2, m is equal to 3, m is equal to 4 and so on and so forth. So, the more the proteins bind, the energy is less. So, according to this energy concept, the system would like more and more protein, all the proteins to bind. If there are 4 proteins, if all the 4 protein binds, the energy will be the least. Okay. So, but what about, but we said that energy is not alone the thing that matters. What matters is energy minus temperature into entropy, some quantity called free energy, that is what it matters. So, entropy also plays an important role in deciding what is the equilibrium of the system, how many proteins will be binding. So, we have to also calculate what will be the entropy. So, that is the thing that we are going to calculate, entropy. So, before that, let us think about this a minute. Here this energy is m epsilon kBt. So, let me divide this by n, energy per, there are n proteins, energy per protein is minus m by n epsilon kBt. So, m by n, there are n binding sites, out of that m is occupied. So, what is this mean? This is basically the fraction of sites that is bound. So, the m by n will be the fraction of sites that is bound. Here m actually means the number of sites occupied, okay, number of proteins bound. M is here basically the number of proteins bound. Okay. So, there are n binding sites, m proteins bind. So, m by n is the fraction of proteins bound. So, let this fraction we call it rho. So, if we can call this as density or fraction, this, so this will become minus rho epsilon k b t. So, just to remind that we can rewrite this in a simpler fashion and we will use this. So, this rho is basically a density, that means how many proteins are bound out of there are n binding sites, the fraction of proteins bound, that is what it is. Simplifying, we are rewriting it in a simple manner, so that we can use rho in a little more easier manner. Okay, now, we have to think about entropy. So, we said that energy is not enough, if by just knowing about energy would not be sufficient. Energy will only tell you some part of the story, and the real thermodynamic equilibrium is decided by the free energy. And the free energy, as we said, is F is E minus T s, same as G, which is H minus T s. H is also energy only, it is written in a different form. So, we will use this here, but does not matter, exactly the idea is same. You can you have to subtract this part, like you have to calculate entropy, and this is then only you will get the free energy, and free energy is what decides the equilibrium of the system. So, let us calculate what is entropy. So, we said that entropy is nothing but law is related to log of the number of possible arrangements. So, if you can count entropy, we just discussed how do we calculate entropy. The way to calculate entropy is count all possible arrangements, it is just simple counting. If there are 5 possible arrangements of proteins, then entropy will be kb log 5. So, we said that S entropy is kb log omega, which is the number of possible arrangements. Now, let us think about what are these arrangements here. So, we have DNA here. So, let us think about this in a very simple manner. Let us say there are 3 binding sites and 2 proteins. So, 3 binding sites and 2 proteins. How many of them will bind? And so, let us say, so we have this DNA and the binding sites are, let us say, this is one binding site, this is another binding site, this is another binding site. Now, what are the possible arrangements? So, let us think of this. So, you can have protein bound all the 3, there are 2 proteins only. So, the proteins can bind here and here. So, let us say out of this 3 places, 2 of them proteins bound. This is one arrangement. Another arrangement possible is this is bound here, this is bound here, this is free. This is the third arrangement, this is another arrangement possible. Out of these 3 binding sites, 2 of them are bound by proteins. Another arrangement possible is, like out of this 3 arrangements, there are 3 binding sites. Out of this, these 2 are bound. This is one arrangement possible, where the first site is not occupied. Is there any other arrangement possible? So, we listed 3 arrangements here. These 2 are bound, the last one is free, here the middle one is free and the other 2 are occupied, here the first one is free and the other 2 are occupied. So, this is if we have 3 binding site and 2 proteins, there are 3 arrangements possible. So, omega number of arrangements here is 3. Now, think about a little more 4 binding sites and 2 proteins. So, let us think of 4 binding sites. There are 4 binding sites and there are 2 proteins. So, then we can have this, we can have this, we can have an arrangement which is like this, we can have an arrangement which is, so this here. So, this is one side, we can have an arrangement this, and we can have an arrangement this, and we can have an arrangement so there are 2 arrangements in this particular manner possible, and we already listed this arrangement, PP arrangement we already listed. So, this is second, then you can have an arrangement which is this and this. So, we now had 1, 2, 3, 4, 5, 6 arrangements. Any other arrangement possible? So, the protein can bind on second side first here, then here. So, for these 2 sides are empty, this and this is empty, are occupied, these two are occupied. This is a possible arrangement. Then here this is a possible arrangement, this is a possible arrangement, and this is a possible arrangement. So, I think only 6 arrangements are possible. So, here omega is 6. Now, think about there are 4 proteins and 4 binding sites. If there are 4 proteins and 4 binding sites, there is only one arrangement possible, all binding. Then omega is 0, omega is 1, and entropy Kb log omega is 0, because log of 1 is 0. So, in this way we have to count. So, the question is, if there are n binding sites and out of them only m of them has to be chosen at a time, how many such arrangements are possible? And the answer to that is NCM, it is in a combinatorics. You may have learned in school that in such cases there are NCM possibilities. So, NCM is n factorial divided by m factorial n minus m factorial. We studied during probability this NCM, n factorial divided by m factorial by n minus m. So, this many arrangements are possible. So, here is say that total number of arrangements possible when there are n binding sites and m proteins bound. So, there are m, if you want m proteins to bind, so m bound proteins, that is what I mean here, there are m bound proteins, then this many arrangements are possible. So, this is omega, then the entropy has to be Kb log of omega. So, this is the entropy, S is equal to Kb log omega, so which is Kb log n factorial by m factorial into n minus m factorial. Okay, so, now what is this? Can we say some? So, this is our entropy. If we know m, if we know look at here, if we know n, if you know m, n minus, if you know n minus m can be calculated and this number can be calculated, a log of that can be calculated. So, the entropy can be calculated. So, for each n and m we can calculate entropy. Now, we get entropy, we get energy when we can calculate the free energy. So, let us rewrite this in a simple manner for simplicity. So, let us say, so let us calculate this log of n factorial by m factorial by n minus m factorial. So, let us write here log of n factorial by m factorial n minus m factorial. So, log a by b is log n factorial minus log of m factorial into n minus m factorial. Log A by B is log A minus log B. Now, this is nothing but log n factorial minus log of m factorial. Log A into log B is log A plus log B. So, log n minus m factorial. So, we can rewrite this, and it turns out that there is a log n factorial can be easily calculated by, if n is very large, there is an approximation. So, there is something called Stirling's formula, which says that log n factorial is n log n minus n. So, log n factorial can be approximately equal to n log n minus n. So, if you rewrite this, what would you get? So, this is n log n minus n log n minus n, that is this term. Here is m log m minus m minus m log m minus m, and here is n minus m log m minus m minus n minus m log n minus m. Now, this is this. So, then you can see that some terms cancelling. So, there are few terms that will cancel from this, because there is an and you can rewrite this, and we can use the definition that m by n is, we define m by n as rho. So, if we define m by n as rho, so let us say we define m by n is basically the fraction of the sites that is bound by protein. So, this is fraction or the density of sites that is occupied by proteins. Fraction of sites that is occupied by proteins. So, that is m by n, and if you use this definition, S can be rewritten in this particular way. S can be written as minus k b n rho log rho plus 1 minus rho log 1 minus rho. So, we can rewrite it, and you can see that this is the entropy. So, we said that the energy is m epsilon k b t, which is minus n rho epsilon k b t. You can write in terms of rho here. So, energy is, there are m proteins bound, each protein has an energy epsilon, binding energy minus epsilon. So, minus m epsilon k b t is the energy, and there are which can be written as m can be written as n rho, and because we saw that m by n here, if you look at here, m by n is rho. So, m is n rho. So, we can write this as minus n rho epsilon k b t. So, this is energy and this is entropy. So, the free energy is E minus T s, which is this entropy part, this is energy part minus plus entropy, minus will become plus. So, this is the total free energy. We want to find the minimum for what. So, the question we have to ask is that for what value of rho this will be minimum. If you, for what value of rho, for what, how many, when, what fraction of this bound, for rho is basically m by n. So, rho is essentially directly proportional to number of proteins bound. So, for what value of rho this f will be minimum? So, that is a question we have to ask, and that will be the rho for which, that will be the equilibrium value of rho. So, that is what we want to find out, and we can immediately imagine that if we have a function f, which is a function of rho, by calculating del f by del rho and equating to 0 will give you the minimum of f. We learned this in calculus. So, this is what we are going to do now. We will, before doing it, we will plot this function and see how does it look, how does the free energy function looks like. So, we will plot this function and see the free energy function. So, before plotting, let us do some rearrangement. So, look at here. What we have here is free energy in terms of there is an n here, there is a kBT here, there is an n here, there is a kBT here. So, I can divide throughout this by n and kBT. So that you can get f by n kBT. So, I can divide throughout by n and kBT. So, that I get f by n kBT. What does this mean? F by n means free energy per protein, there are per binding site, that is what f by n means, and in unit kBT means in units of kBT. So, this is a dimensionless number. So, this will be energy, this kBT is also energy. So, this is just for simplicity we will make it some quantity which is essentially free energy only, but we will write in units of kBT. So, we will it's easy to plot it if we had f by n kBT. So, we will plot f by n kBT versus rho. So, this is what we similarly. So, then if you write f by n kBT, what you get is minus rho epsilon plus rho log rho plus 1 minus rho log 1 minus rho. So, this is the free energy per binding site in units of kBT. That is what this is. Now, if you plot this, what you get is this. So, look at here carefully. The red line is the energy, the green curve is entropy, and the blue curve is the free energy, which is this curve minus t times this curve. So, f minus T s is what this is. Okay. So, this is energy. So, this is minus, we said the expression for energy, we said this, we discussed the energy expression, which is basically this one minus n rho kBT, and you are plotting as a function of rho. So, what you would get here is this, and here this is entropy, this is our expression. So, we had some expression. So, we have energy which is minus n rho kBT and entropy which is rho log rho plus 1 minus rho log 1 minus rho. So, these are these two curves. So, if you look at here, what does this mean? This means that according to this, energy is minimum when the protein density, that means the fraction of bound protein is 1. That means, everything is bound, the protein density, when the density is 1, the free energy has its least value. The energy has its least value. E, the red curve is least when this is 1. On the other hand, entropy is maximum somewhere in the middle, roughly half of them, like somewhere in the middle is entropy is maximum here. Entropy is a function which is like, looks like this. Here is small value, here is also small value, somewhere in the middle is entropy is maximum. So, entropy wants rho is equal to 0.5 or somewhere this, while energy wants rho equal to 1. And so, entropy and energy compete, and the free energy will have a minimum somewhere in between. So, the minimum here is somewhere close to 0.9, somewhere here. This is the minimum of free energy. So, by just looking at the plot, we know that energy is minimum here, entropy is maximum here, and the free energy is this. So, here for a particular value of epsilon is what is plotted here. For epsilon is equal to 2 is this plot is 4, and epsilon is equal to 2, this plot is, so this is this vertical line represents the value of rho, for which, for the pink line represents the value of rho for which the entropy is maximum. The black line, the black vertical line represents the value of rho for which the free energy is minimum. So, it turns out to be around 0.88, this is the value. So, and this 1 is the value for energies, for energy the E is least. Now, we want to find out the some general formula for this minimum of free energy. So, we said that if we have an expression f of rho, the free energy minimum can be calculated by calculating the derivative del f by del rho equal to 0, and we said that the f, so we will do this f by kBt. So, let us call this f tilde, we will do this f by n kBt and minimize this, which is also fine. So, which is basically minus rho epsilon plus rho log rho plus 1 minus rho log 1 minus rho. This is our f tilde. So, this is a new f. So, now if you want to calculate the minimum of this, we have to find the derivative of this. So, let me call this quantity as f bar, this whole quantity as f bar. So, this quantity as f bar. So, I want to find out del f bar by del rho. So, the first derivative of this will be epsilon, and rho log rho will have a derivative. Rho will be this, log rho will have a derivative 1 over rho plus log rho, because derivative of rho will be 1 and there is a log rho, and similarly here you will have 1 minus rho by 1 minus rho plus log 1 minus rho. So, what is this? And there is a minus sign here, because we are taking derivative of rho. So, there is a minus sign here. So, what is this? If you calculate this, what you will get is that minus epsilon. So, del f by del rho, del f bar by del rho here is minus epsilon, and this is rho by rho is 1, and 1 minus rho by 1 minus rho is also 1, and there is an opposite sign. This and this cancel, 1 minus 1 will become 0. So, you have ln rho minus log 1 minus rho. So, you have plus log rho minus log 1 minus rho, and this has to be 0. This means that epsilon is equal to log rho minus log 1 minus rho, which means epsilon is equal to log of rho by 1 minus rho. So, there is a relation between energy and density, and you can invert this and write that e power epsilon is equal to rho log 1 minus rho. So, this would mean that e power epsilon. So, let us exponentiate this. E power epsilon is equal to rho by 1 minus rho So, in other words, this can be also rewritten as this particular way, rho is equal to e power epsilon by 1 plus epsilon. So, it turns out that if you know a value of epsilon, the rho for which del f by del rho will be 0 is same as is given by this formula. So, when rho, if epsilon, let us say if epsilon is 0, that means there is no particular energy, binding energy. If epsilon is 0, then e power 0 plus 1 plus e power 0. So, when epsilon, let us look here, when epsilon is 0, this rho is basically e power epsilon by 1 plus epsilon, 1 plus e power epsilon, which is 1, e power 0 is 1 by 1 plus 1, which is half. So, when there is no particular energy preference, only entropy, then half of the sites only will be bound. There is no energy 0. If energy, binding energy is, let us say, infinity, that means all proteins are bound, they all would like to bind, like if epsilon is infinity and the binding energy is minus epsilon k b t, then if epsilon is infinity, so that the binding energy E is minus. So, we said that the binding energy is minus, for one molecule is minus epsilon k b t, one protein. So, if epsilon is infinity, the binding energy is minus infinity k b t. So, that means by binding infinite energy will be gained. What does this mean? That everything would love to bind, because there is infinite energy gain. Then this energy will dominate over everything else. So, if you put here in this formula epsilon is equal to infinity, you will get rho is equal to e power infinity by 1 plus e power infinity. E power infinity is infinity, which is, so this will be a large number and this will turn out to be 1. Okay. So, the answer will turn out to be 1. And if epsilon is minus infinity, that means if the binding energy is plus infinity, that means it does not want to bind at all, proteins do not want to bind at all, then the rho will turn out to be. So, let us say there is another case, is another limit is epsilon is minus infinity. So, that the binding energy is minus epsilon k b t is equal to infinite k b t. That means, the binding energy is infinity, that means it does not want to bind at all. Then the rho will be e power minus infinity divided by 1 plus e power minus infinity, and this will be, this is 0 by 1 plus 0, which is 0. So, if the binding energy is infinity, no proteins will be bound. So, this will give us the correct, the binding density, the how many proteins. So, we can predict using ideas from thermodynamics how many proteins will be bound on the DNA. So, for epsilon is equal to 2, here is what I have plotted here. This will give you the protein density bound. So, for epsilon the free energy minimum is around 0.88. So, 88% of will be bound when epsilon is equal to 2. So, this is one use of knowing thermodynamics. We can predict what will happen in various biological contexts, various systems. So, this is a simple example, in vitro example we discussed, and the same ideas can be applied to little more complex situations and studied. We will now discuss another example which is very similar to this, is basically DNA melting. So, you all know that you increase the temperature, DNA will melt, double standard DNA will melt. And can we predict at a given temperature what will be the equilibrium state? It will be melt, or it will be completely melt, or most of them melt, completely they would have formed single stranded DNA, or is it the case that they all will be base paired? It turns out the thermodynamics, our simple ideas that we learn in thermodynamics can predict this. So, let us think of a thought experiment. So, this is a very simple thought experiment where we can do all the calculation paper, and you can think of how to extend this to reality. So, let us imagine that a 3 base pair double stranded DNA. So, let us say you have double stranded DNA, 3 base pair. So, now you can imagine some situations, like you can imagine that this is all the three are base paired. You can imagine a situation where let us say only two of them are two contacts are two base pairs are broken and there is only one contact. So, you can imagine this kind of a situation where these are broken, or you can imagine another. So, let us consider two cases. So, case 1, case 2. So, case 1 is all base pairing is intact, like all this there are bonds, and here there is only like one bond and two other base pairs are broken. Now, we want to calculate the energy, free energy for case 1 and case 2, and let us calculate the free energy for this case 1 and case 2, and let us see what do we get. So, what is free energy? Free energy is energy minus E minus T s. So, this is our free energy F or G. Now, what is E? So, let us assume that E is equal to, just like we did, some value, let us say H. So, H is some value which can be 100, 200, 300, some value like kBT in units of this kBT. There will be some energy, let us call this H. So, for each base pair the energy is H. H is basically energy per base pair. So, if there is a bond is there, that will gain an energy H. So, this has an energy 3 H. This is an energy H, case 1, because there is only one bond here. So, let us call this an energy H. Energy H is the energy per base pair. If there is one base pair, that is the energy gain of H. You have to give. So, and if there are 3 base pair, the energy is 3 H. Now, what is entropy? So, let us think of these two cases again. So, we have two cases. First case is all base pair intact, and the second case is two base pair broken and one is intact. And this we found that this is an energy 3 H, and this is an energy H, because only H is basically energy per base pair contact. If the contact is one base pair, this is 1 H. Now, we have to calculate entropy. So, what is entropy for this case? Let us think about it. Entropy for case 1. So, case 1, 3 base pair intact. This is our case 1. How many states are possible when the 3 base pairs intact? There is only one state possible. This is 3 base pair intact, there is only one state possible. So, the omega is 1. So, entropy is Kb log omega. So, this will be 0. So, free energy is E minus Ts. This is H minus 0, which is just H. So, H kBT is the free energy in units of this. Now, similarly, we can calculate entropy for this. The 3 cases possible for this. So, think about it, such a state is possible. You can have this and this two broken, this is another state possible. You can have this is broken, and the middle one is 3 states possible. So, the entropy for this is Kb log 3. So, log 3 is roughly 1. So, this is just Kb, entropy is say Kb. So, the free energy for this case will be, so for the case 1, the free energy will be just h kBt, case 2 will be free energy is 3 h kBt, because there are 3 bonds and this is the energy and entropy is 0, and the case 2 free energy is h kBt minus just kb log 3, which is kBt. Okay. So, this will be the answer, kb log 3. So, now you can calculate basically what will be the free energy. Now, you do this is an assignment for you, vary temperature, take T is equal to 100 Kelvin and T is equal to 1000 Kelvin, and see which of this is minimum, and take H is equal to minus 100 in this cases. So, for when T is equal to 100 kBt, so and T is equal to 1000 kBt, this is an assignment for you to do, see that which of this will be minimum, and you will see that the state where the free energy is minimum will be the state at higher temperature. You will find that the state where all base pairs most of the base pairs are broken will be the minimum energy state, and at lower temperature all intact, the more number of base pairs will be intact. So, this is something which we expect, and this we can see from our simple calculation. Okay. So, the idea here is that we can go ahead and use this kind of ideas for many cases. So, to summarize what we learned today is how do we think about calculating energy for two systems. One is proteins bound on DNA, and ask the question at equilibrium how many proteins will be bound, what fraction of proteins will be binding sites will be occupied, can be answered by using thermodynamics. Similarly, you can also answer the question what will be the state at a given temperature such that the free energy is minimum. So, that will give us the whether the DNA will be half melt fully, what fraction of the DNA will be base pairs will be broken when at a given temperature T can be also answered by this. So, with this I will stop today's lecture.